For 20 years, GMOs have spread across the planet. Genetically modified organisms. They're predominantly plants, their genetic material transformed by scientists primarily to increase their yield. Soy, corn, cotton, canola. GMOs are now said to cover over 10% of the world's farmland. But they're also the object of fierce opposition.
We head north and we stick together. We're here to chop down crops and assert the rights of all citizens to refuse GMOs.
In Europe, the vast majority of the public is opposed to genetically modified crops. All over the world, people are concerned about the dangers of GMOs, not only for public health, but for the environment and for farmers as well. They criticize the influence of the seed and pesticide giants like the American company Monsanto.
Monsanto, dégage! Le peuple rat à peau! Le business, dégage! Le peuple rat à peau!
Imminent disaster for some, major breakthrough for others. The global controversy between those who are for or against GMOs has made for a heated and sometimes obscure debate, polluted by false arguments that have no real basis in science. How do we separate truth from false? What are the facts about GMOs?
Small piece of dry ice. And let's see.
The United States is where the first GMO crops were grown. Bt crops, which belong to one of the two major categories of GMOs, were designed to resist harmful pests like the corn borer and rootworm.
Ah, here's one. The rootworm beetle is often referred to as the billion-dollar bug. That is a low estimate of the cost. And the $1 billion refers to the losses caused by the insects and the money spent to try to protect corn from rootworm beetles. When we have a lot of beetles, they can lay so many eggs in there that an unprotected cornfield would be practically destroyed. The larvae feed on corn roots in order to survive. Let's see if there's any larvae in here. We're going to bring it out and put it on the tray. One of the things you can notice about this root is all these fine root hairs, all this fine tissue here. If there were a lot of rootworm larvae, they would have chewed away all this fine root tissue. Roots that are not protected from rootworm beetles and that are fed on heavily will look like this. This one was not protected either by a soil insecticide or any sort of Bt corn. The GMO plants were constructed by inserting a gene from the soil organism Bacillus thuringiensis.
Bacillus thuringiensis, or Bt, is a bacteria that secretes several insecticidal proteins. To create a Bt plant, scientists perform transgenesis. This involves extracting a gene from the Bt bacteria, then inserting the DNA fragment into the plant's embryonic cells. This transgene, derived from another organism, produces a toxin that will be ingested by insects if they eat the plant.
It opens pores in the insect's midgut, in its intestinal system, and the larvae would die. It's targeting receptors that are only present in the insects. We don't have these receptors. And one of the great advantages of Bt corn is that it only kills the target insect. Unlike when we spray a field with insecticide to try to kill adults, we kill all the insects that are in the field. It has an added advantage in that the growers are using less pesticide, but Bt corn costs more than regular corn. However, these Bt corn varieties are producing much greater yields than we used to expect. And so the profitability is there.
The other main category of transgenic plants is called HT, herbicide tolerant. Over 80% of GMOs grown today in the world contain this trait, especially soy. Once the plant has been injected with a microbial gene, it is able to survive a broad-spectrum herbicide like glyphosate, the active ingredient in Monsanto's notorious Roundup. This means that when farmers spray Roundup onto their fields, all the weeds will in principle be destroyed, but not the transgenic crops, which tolerate Roundup and are called Roundup Ready.
1996 was the first year. When we first came up with the Roundup Ready soybeans, we were ecstatic because I think there wasn't a farmer in the country that said, man, if I could just have a soybean that I could spray Roundup on and it would kill everything except the soybeans, and then all of a sudden there it was. And it made controlling the weeds so easy. So it was almost a total and complete adaptation to the GM crops. Never in the history of the U.S. in agriculture had any technology ever been adapted so fast and so completely. There was not a yield boost. It was just easier. And you could farm more acres, so you didn't have to make as much per acre. You could just farm more acres and make fewer dollars per acre. So it pushed that revolution of, you know, you got to get bigger, you got to get bigger, you got to get bigger, you got to farm more acres. This is our farm on this side. This is about 3,000 acres, or is not a huge farm. The next farm, the guys that farm the next farm to us, farm 8,000 acres. That's why you see, we'll drive for miles and you see no houses anymore. And there used to be 6 houses with people when I was younger, and now they're not. I mean, the houses are tore down. This house, it's vacant, so you'll notice not a lot of traffic.
Today in the United States, GMOs represent around 90% of soy, corn, and cotton agriculture. In Argentina, it's nearly 100%. Argentinian farmers have overwhelmingly adopted GMO technology. This is particularly true for GM soy, grown and harvested in vast plantations.
Soy had a boom, first because compared to other crops, it's easier to handle in terms of applying herbicides, fungicides, etc. Next, because we got good prices on the international market, so it was a profitable crop to grow. We devoted many fields to soy production. The size of these fields is impressive, but that's the kind of acreage we're looking for in our production sector. Huge surfaces that will allow us to dilute costs.
Since the introduction of GMOs in 1996, the land area devoted to soy has tripled in Argentina, reaching a total of nearly 20 million hectares today. The intensive monoculture of soy has pushed back other crops, along with the forests. The legendary landscape of the Pampas, dotted with cows roaming free, has given way to feedlots. For supporters, GMOs are an essential tool for meeting the food needs of a rapidly growing global population, which is set to increase from 7 to 9 billion in 2050. It's the classic argument of GMO companies like Monsanto, the global leader, which makes $15 billion in turnover and employs 20,000 people in around 60 countries.
This type of crop allows us to obtain a better yield per unit area. This is how we're going to feed the world of tomorrow. The new technologies we're currently rolling out increase average yield by 7 to 10% for soy. Extrapolated to the entire country, that's considerable. This year, we've seen soy yields of 8,000 kilograms per hectare. It's huge. And what really helped Argentina pull itself out of the crisis was agricultural production. In fact, we're one of the major exporters, not just of soybeans, but also of oil and flour. It's the country's biggest source of foreign exchange.
Many countries, shown in orange on the map, import GMOs, including Europe for livestock feed. And 28 countries, shown in green, grow GMOs. But of the 180 million hectares farmed in 2014, 85% are concentrated on the American continent, and 40% in the United States, the leading producers of GMOs on the planet. Transgenic crops have now become the norm on American farms. And in laboratories, new plants are being tested. Like potatoes, genetically modified to resist diseases such as late blight or the potato virus Y.
We're here in the tissue culture lab where my postdoc here, Swati, is doing genetic engineering of the potato. So it's very simple. What we do is we take these plants and we cut them into pieces, the leaves and the stems, and then those can be used for the gene transfer process or the transformation process. But in the other part of the lab, we're growing the bacteria that has the gene inserted into the plasmid of the bacteria. And so we create the wound surface on both sides of the leaf so that the bacteria can infect that cell, and in the infection process, we'll actually insert a small segment of DNA that we placed in the bacteria. And so if the transformation was successful, we would expect to see some shoots regenerating. And so if we're able to put resistance genes into the potato, for example, the late blight disease, we can significantly reduce the amount of fungicides that are applied to the potato. And so having plants that are innately resistant is better for the environment. We know that there's a lot of people that have, concerns about the technology, the safety of the crop. But we live in a world where we can never ensure complete safety. And so we really need to move past that and make this technology acceptable.
Concerns about the potential dangers of biotechnology go back a long way. They surfaced as early as the first GMOs, which were not of plants, but of bacteria. In 1972, American biochemist Paul Berg of Stanford University made the first attempt at transgenesis by recombining DNA molecules in the Escherichia coli bacteria genome. In response to concern over new techniques of the manipulation of living material called genetic engineering, Berg organized a conference in Asilomar, California in 1975, which was attended by 100 biologists from around the world.
Many scientists, including those who were creating GMOs, were very concerned. They were very concerned about what would happen if humans came in touch with these bacteria, but also if these bacteria were released into the environment. So they said, we take responsibility for what's happening in our labs, and we leave it to the future for someone else to take responsibility for environmental releases. But there has never been an Asilomar conference for environmental releases. There was never an evaluation of what could happen.
The controversy over GMOs really began in the 90s, when genetically modified crops suddenly began to arrive in France from the United States. And Greenpeace launched a huge campaign. An article in the national paper, Liberation, really set things off. It was called Mad Soy Alert. A boat with a cargo of genetically modified soy landed in Europe. On the photo, you see the guys from Greenpeace wearing white lab coats and the X from X-Files, which was the series everyone was watching back then, and had a fondness for conspiracy theories. Why mad soy? Because we were right in the middle of the mad cow disease. Mad cow showed us that the agricultural system was taking reckless risks. People had kind of lost faith that institutions would protect them in terms of what they ate and how their health was handled. And right then, GMOs arrive. I think that if there hadn't been that conjunction, there probably wouldn't have been such an intense controversy.
As the debate heats up in Europe, doubts are beginning to be voiced in the United States, right in the heart of GMO land. Because the insects that were supposed to be eliminated by Bt crops aren't behaving exactly as planned. For the past few years, they've started to become resistant to the toxin produced by the plant.
Western corn rootworm resistance to Bt corn is documented now from several states. Began in Iowa in 2011. It's been documented in this field 2 years ago. I think the main reason that the resistance evolved has to do with the way that the product was used. It was such a wonderful product that growers used it year after year after year, and so we overused Bt and we created a population that evolved resistance. And at the time Bt hybrids were commercialized, the expectation was that we could manage these insects without putting tremendous amounts of broad spectrum insecticides into the environment. What's ironic now is that as we face rootworm beetles that are resistant to Bt, we're now putting soil insecticide in some cases on Bt corn hybrids, which is a terrible strategy.
Today, it's not just the farmers of Bt crops who are worried. It's also the farmers of HT crops, crops that can tolerate herbicides like glyphosate, Monsanto's Roundup. Because weeds are also developing a resistance, not to the transgenic plant in this case, but to the herbicide associated with it.
There's another one.
These superweeds, as they're called, are now prevalent on American farms, even more than super pests.
You can see the lighter green are what we would call the water hemp, which we have a much more significant problem in this field with that. And it's not just us, it's everywhere. You can see where the spray has been sprayed on it when it was younger, and the green that's coming back. And on the mare's tail, you can see how it affected the plant, but it didn't kill it. And it certainly, we call that, we just made it mad. And each year it only gets worse. So Mother Nature will adapt to what's trying to eliminate her, I guess.
We call it Darwinism, and that is the idea that if you push a population with selection in one direction, there will be changes to adapt to that pressure and survive in it. The way it works is that you kill many of the descendants, except the ones that will survive the given pressure, selective pressure, and then those will have more descendants in the future generation and the next generation. Can we say that the GMO technology has increased the phenomenon of herbicide resistance? I think the answer is yes. The introduction of the resistance to one herbicide made it possible to apply much more herbicide any time of the year, so the window of selection of the weeds became much wider through the year and over many years. This selection was kept. So the specialization in one herbicide became a very important thing.
In the late 90s, when Roundup Ready soy was offered, the rate of application of glyphosate per acre was 8 ounces. Today, people...
8 ounces per acre.
Per acre, which is a small amount.
Yeah.
And today, it could very easily be quadruple, asking people to put on quadruple to hope that they can kill these resistant weeds. So, you know, and then the other way we fight it, if you're going to fight it with technology, is certainly the industry's done pretty well on it, because they say you don't just use one mode of action to kill this now. You need 2 or 3 modes of action out there on that soil to kill this plant now. Because if one doesn't do it, you need to get them with another.
Meaning atrazine 2,4-D?
Right. It could be atrazine, it could be 2,4-D, it could be other modes of action. And it's not really a problem for them.
No, it's a business opportunity.
It's sales opportunity,
yeah,
exactly.
Of course.
So you sell more chemicals, so you get farmers to put more on, and so then
they'll never say, sorry, we were wrong. We're going to move out, and good luck, guys.
Yeah.
You know, for a long time, for about 50 years, the industry was moving forwards, always coming up with a new chemical that was a little bit better, less toxic in the environment, more selective, and all that. But they stopped worrying about that. And now what they're doing is going back to the chemicals from the 40s and 50s that we know are really bad.
Contrary to initial promises, the use of herbicides has not decreased with GMOs. Quite the opposite. In Argentina, where herbicides are sprayed onto fields partly by plane, sometimes very close to people's homes, this raises serious public health concerns. According to official statistics, farmland has increased here by 50% in the last 20 years. The volume of phytosanitary products sold, however, has increased by 858%. Since 2010, a professor of medicine has been coordinating a vast epidemiological study in the major GMO farming provinces. The study is conducted by students in their final year.
As of today, our study of epidemiological data involves 21 communities in the provinces of Santa Fe, Entre Rios, Cordoba, and Buenos Aires. In total, there are about between 115 and 120,000 people covered by our study. And we're getting a set of statistics that we find worrying. The average rate of new cancer cases is 386.2 per 100,000 local residents. That's 1.8 times the national rate for cancer cases. We've also noted causes of mortality. And we can see that in most of these communities, cancer has become the main cause of death, even though in our country, the leading causes of death are road accidents and cardiovascular illness. We've also found more cases of birth defects, of children with a disease called cyanomelia. It's a syndrome caused by the defective expression of the COX and COX2 genes, which prevents the proper development of the lower limbs. And when you study the timeline, that is, the date of conception of the mothers of these children, and the stages of gestation, It appears that the first 3 months of the embryo's development coincide with the peak of agrotoxin use in the preparation of fields for GMO crop farming.
In neonatology, which is my area of expertise, there are periods of the year when the intensive care unit is completely full. 90 to 100% of the patients are children with birth defects. This did not used to be the case in Argentina. Women have spontaneous abortions more frequently during the fumigation periods. This rate is 3 times higher than average. It's very clear that this problem appeared and developed with the arrival of genetically modified soy and its agrochemical package. Argentina has between 12 and 14 million people who are fumigated, who live in areas exposed to agrochemical products. That equals 1/4 of the population. There's a fleet of 1,100 planes that represent the Air Force of soy. Aerial fumigation contaminates the water and the air. We found that when it rains and we collect rainwater, it contains a high concentration of glyphosate. We demand that these fumigations be outlawed. For us, it's a criminal practice.
In 2012, a pilot and a GM soy producer were convicted of contaminating a neighborhood of Cordoba, the second largest city in Argentina. The verdict? A 3-year suspended prison sentence. It was a first in Latin America and was due to the determination of local mothers, in particular Sofia Gatica, who received the Goldman Environmental Prize for her work.
It all began in 2001 in the Ituzaingó Anexo neighborhood. I began investigating in the neighborhood because my daughter died of a kidney malformation, and one of my sons couldn't walk after the fumigations. I went from house to house, and I realized that in my street, 6 mothers had lost their babies at birth. Others had leukemia, and nearly everyone I surveyed had cancer. So we decided to put pressure on the government and get some answers. The neighborhood was declared uninhabitable in 2005. The main problem Argentina is facing isn't agrochemicals. It's genetically modified organisms, transgenic crops, because they come with glyphosate and all the agrotoxins.
It's pesticides that kill, not GMOs themselves. But since the appearance of resistant transgenic crops, the use of pesticides has become even more intense and extended throughout the year. In September 2013, despite death threats and several assaults, Sofia Gatica joined a number of residents of Malvinas, a small town near Cordoba, in blocking the construction of a huge Monsanto factory, announced as its biggest factory for GM seeds in Latin America.
Compañeros, pedimos que nos acompañen a esta lucha, que es una lucha de todos los argentinos. Te lo digo, te lo canto, ¡fuera Monsanto!
Te lo digo, te lo canto, fuera Monsanto. Te lo digo, te lo canto, fuera Monsanto. Te lo digo, te lo canto, fuera Monsanto. Sofía,
We've heard that there are certain concerns about the risk. In truth, the risk is non-existent. All the agencies of the countries where the product has been approved have confirmed that these approvals are legitimate and have done tests that conclude that the risk is practically nil. It's very difficult to find studies on glyphosate that show any kind of toxicity or of carcinogenicity.
And yet, that's precisely what appeared on March 20th, 2015, when the International Agency for Research on Cancer, the specialized cancer agency of the WHO, published a scientific evaluation of 5 pesticides, including glyphosate, the world's top-selling herbicide. The summary was printed in the respected journal The Lancet Oncology. A monograph presenting all of the scientific studies that were reviewed will soon be published.
The evaluation process takes about a year. We assembled 17 experts from 11 countries, and approximately 400 studies will be cited in the final monograph. We have 4 different levels of classification. Group 1 are things like cigarette smoke, asbestos, where we're fairly sure the human evidence is knowing that these are causing cancer. Group 2A is one step down from there. This is strictly a very narrow question of, does the agent cause cancer? And in this case, our working group concluded glyphosate is likely to cause cancer in humans.
We don't know which studies they used because the International Agency for Research on Cancer's monograph hasn't been published yet. Other agencies, like Germany's Federal Institute for Risk Assessment, which reviewed more than 55 studies over 5 years, concluded on the contrary that there is no link between glyphosate and cancer.
The study by the German Institute is a separate evaluation, and it's done according to a different procedure than ours. There are several differences. For instance, as I mentioned, our working groups are experts who are clearly independent. We would not have somebody who was an employee of a company who was manufacturing a substance be part of a scientific evaluation. They can come to the scientific evaluation as an observer, but they don't have a voting role. They aren't drafting things for our working group to consider. They have a hands-off role, and that's not exactly the procedure that the German government, for instance, follows. It definitely had a big impact. I think it was unusual in this case because it was one particular company that that really was making a lot of media stories and so forth, and contacting us and so forth.
Do you mean there is some pressure?
There can be pressure to make change, pardon me, but excuse me. But we will not be making any changes due to pressure.
It's a plane coming, about to spray us.
The increased use of pesticides, which is linked to the resistance phenomenon, raises serious public health concerns. But there's another phenomenon of equal concern, which targets biodiversity, the risk of contaminating traditional crops that are grown near GMOs, a risk long denied by GMO companies until scientists revealed exactly that in Mexico.
I come from Mexico. I was raised there, and I've worked there for many years in collaboration with some indigenous communities in southern Mexico. And we found, almost by accident, not really expecting it, that there was positive GMO DNA within the corn plants of the Oaxaca people. There had been cross-contamination. There has been what's called inbreeding or introgression of the GMO DNA from the GMOs into local plants. And our results have been published in Nature magazine. So it became immediately a big source of controversy and a headache for the industry. Monsanto was saying that a distance of 1.5 meters was enough to prevent cross-pollination between a plant of corn here and a plant of corn here. And that, I think, to anybody was just not believable. So up here, you have the tassel where the male flowers are produced. And this pollen moves by wind, gets attached to a silk. So one single pollen grain comes into a single silk and then starts traveling down until it finds the ovary of the female flower and fertilizes it. And so each one of these kernels is really carrying the genetic material from both the father, the male flower, and the mother. Monsanto had argued at the beginning that they had control. And what our paper did was to show simply that they didn't. That you cannot really control something that moves through the air. And the whole situation became very, very complicated for me because immediately we started seeing a discreditation campaign. I was very close to losing my job. I had to fight for about 8 years because there were many people telling my university that I should be kicked out for doing this work. And I was treated, gangster style sometimes, that they threaten you and they tell you about your children and they know where they go to school, threats like that. Fortunately, our results have been confirmed by other researchers many times over.
In the case of corn, 95% of the time, corn pollen has dissemination distances of under 100 meters. But there's still 5% that goes into the atmosphere, and that pollen, which is viable, can pollinate non-GMO corn 2 to 3 kilometers away. Pollen, in fact, rises up into the air. Temperatures are cool, there's humidity, and so the pollen remains viable and is able to go to pollinize corn several kilometers away.
The phenomenon of contamination presents a major risk for the diversity of genetic resources, particularly for a country like Mexico, the land of the origin and diversification of corn. In September 2013, after several years of legal battles, a Mexico City district court banned the import and cultivation of genetically modified corn in the country. But Monsanto appealed several times, and in the end, the ban was lifted in 2015. If contamination is a concern, it's also, and maybe especially, because GMOs aren't regular plants. Seeds are patented, protected by intellectual property rights. This forces farmers to purchase them every year, even though until now they could select and plant seeds from the previous year, which cost them nothing.
I first lost my illusions whenever I saw the patent that was held on the seeds. What the patent did was said, you can no longer retain any for your own use. You have to go back to the holder of the patent to get your seed. And I think what flew in the face of many American farmers, especially my father, is, how many people have ever bought anything and it wasn't theirs? I mean, and he would say, it was our soil and our toil, and then what I grew out of my land wasn't mine. And to have to go back to that patent holder it was basically, for what we would call in the U.S., a cash cow. I mean, to allow a patent on a life form virtually gave a monopoly to the patent holder and no competition in the market. And we saw from the beginning when the first for that trait on Roundup Ready, if you will, $6 for that for that technology per unit, as they called it, going from $6 well up into the mid-20s. It was such a game changer that we shifted all that control over to a few corporations' hands. I believe Monsanto owns about 95% of those patents. I saw how, if you will, Monsanto wanted to enforce their ownership and sent private investigators out and sued the very people they were selling to, farmers who retain their own seeds. They sued a lot of farmers.
In June 2000, Monsanto went even further and sued Canadian farmer Percy Schmeiser, whose conventional canola seeds had been contaminated by Roundup Ready canola. The firm accused Schmeiser of patent infringement for planting these seeds without a license or payment of royalties.
Percy Schmeiser and Schmeiser Enterprises Limited versus Monsanto Canada Inc. et al.
Canadian judges ended up ruling in favor of Monsanto. Percy Schmeiser was declared guilty of patent infringement and quit canola farming.
Let's say you lived in a city and you wanted to paint your house purple. And all your paint, it was very windy, and all your paint blew over on your neighbors' houses. Well, you could sue your neighbors for your paint getting on their houses. That's how the rules were set up for Monsanto.
For me, that was a scandal, a legal scandal. I don't see how the fact that a gene is patented and comes from the crop next door means that I should be robbed of seeds that I take from my own plants. So patenting is something that's extraordinarily dangerous for the diversity of cultivated crops. Because if these companies end up owning practically all human food, there'll be no more diversity in cultivated crops, or very little. Just the few genotypes that are grown by the seed companies. And that is really very dangerous. It would take a single disease to develop on some of these genotypes, and we would have a worldwide agronomic catastrophe. Massive famines on a global scale. I mean, we can discuss whether it's bad for rhinoceroses to exist or not, but the diversity of cultivated crops is simply essential for the survival of the human species.
Stand up for your rights. Don't give up the fight against GMO.
In Africa, the introduction of GMOs and patented seeds has become a very sensitive issue. Only 3 states, in particular South Africa, grow transgenic crops today, but more and more countries are conducting trials, like right here in Ghana. In this small West African country, a number of people are fiercely opposed to this. They've created an NGO with a name that sounds like a manifesto, Food Sovereignty Ghana.
We refuse to be what they wanted us to be. We are what we are.
Its activists took the government to court to try and stop the planned commercialization of genetically modified rice and BT cowpea, a traditional bean.
Fantastic. We need all the support that we can. That's right. We want to kick GMO out of this country.
In 2015, 5th day of February, when Food Sovereignty Ghana went to court, there was no National Biosafety Authority. It means that they have not respected the Act, because the Act said that there should be established a National Biosafety Authority to see to all issues of biosafety in Ghana. They didn't do that. We're sitting down and taking the citizens for granted. So we are saying that everything they were doing is illegal.
Going the GMO way would enslave African populations. It's a form of neocolonialism. So we don't have to tamper with the livelihood of farmers by getting into contracts where we concede their rights to profit-oriented organizations who are going to patent seeds. Some of the seeds that are going to be patented are originally from Ghana. The home of origin of cowpea is Ghana, and it's totally unacceptable to us. We want the government to change the course away from introducing GM crops into Ghana, because we have food that goes to waste in Ghana. We produce more food than we need. but the issue is access to markets and post-harvest loss. So if we have more roads from the farm to markets, we have more warehouses, there'll be no issue of food security.
But in October 2015, their application against GMOs was rejected. The judge considered that the accused, in other words the government, would suffer enormous losses, and this would undoubtedly jeopardize investment and donor support. Near the Gulf of Guinea, like elsewhere in Africa, supporters maintain that GMOs represent an essential tool in ensuring the continent's food security. Not only by improving agricultural productivity and feeding a rapidly growing population that is set to double by 2050, but also to deal with the effects of climate change, by which sub-Saharan Africa is particularly affected. Experiments are therefore being conducted on drought-resistant corn and on flood-resistant rice. From a genetic point of view, these are complex traits, which means that today, in this area, GMO technology is much less efficient than conventional selection methods. But numerous trials are currently underway in Africa, financed by foundations like the Bill and Melinda Gates Foundation or USAID, the United States Agency for International Development. The Americans seem to be quite active in promoting their agricultural biotechnology industry to African leaders.
The foreign policy of the United States has tied the GMO to, what they do. So, before you get some aid, there's some GM coming through there. Sometimes they use nice words like hybrid seeds and seeds that will increase, your yield. But these are all basically boiled down to the same thing. GM has nothing to do with increasing food security. It's rather to promote business. And the same companies are lobbying governments to be able to come in there and change their laws, change the seed laws to control these seeds so that their companies can come in and provide these seeds.
North of Ghana, in Burkina Faso, the government approved the commercialization of Bt transgenic cotton in 2008. Monsanto signed an agreement with Sofitex, the country's leading cotton company, with the idea of turning the country into a model and convincing other nations in the West African sub-region. In 2015, Burkina Faso produced over 700,000 tons of cotton, 70% of which was genetically modified. It became the biggest cotton producer on the African continent.
This year, we produced around 567,000 tons. So, as you can see, we do indeed produce 80% of Burkina's cotton. Now, that's one of the storage warehouses. Cotton's probably 50 to 60% of Burkina Faso's export earnings. It's the country's white gold. During the 96-97 campaign, we had a terrible problem here in Burkina Faso, also in Ivory Coast and in Mali. The pests, that is the insects, became less reactive to the pesticides we were using. This led farmers who usually applied 5 or 6 treatments to sometimes increase it to 12 or 18 treatments, all for an uncertain result. It was a terrible problem, and it was while trying to find a solution that we got in touch with Monsanto.
Here's the biggest cotton pest. They're butterflies. The larvae are very well controlled by the toxins in the GM cotton used in Burkina Faso. It makes the work less difficult because there are fewer treatments to apply, from 6 down to 2. That's 2/3 fewer treatments, with an average yield increase of 30%. As we observed in trials that lasted 6 years, from 2003 to 2009. It brings farmers greater earnings and an easier life.
In Burkina Faso's cotton-growing regions, many farmers were thrilled with the arrival of BT cotton, which allowed them to reduce the number of phytosanitary treatments and free up time for other activities. The people who welcomed it most were farmers with surface areas and harvests large enough to offset the increased cost of patented seeds, which comes to around 40 euros per hectare, as opposed to €1.50 for conventional cotton.
I grow the new cotton, BT cotton. I have other fields, but this one measures 10 hectares. It's better for us to be able to treat only twice, because treatment is unpleasant. Before, when we treated, we were often sick because of all the pesticide emissions. The new cotton also increased our earnings. When we grow it, it produces a lot. On 1 hectare, if we put manure, we can get 2 tons. Whereas before, with the old cotton, despite all our care, we only got 1.5 tons. Since there were many changes and I got some money, I was able to buy this tractor.
Today, however, anger towards GM cotton is building in Burkina Faso. Because 7 years later, some of the initial promises have not been kept. The increased cost of BT seeds was supposed to be compensated by fewer treatment purchases and by higher yields. But the 30% increase never happened. It's closer to 17%. Not only that, but many producers are harvesting lighter seeds than with conventional cotton. This creates losses for them because cotton is sold by weight.
I grew GMOs in this field right here, and I ended up deeply in debt, so I had to stop. When it's growing, you think the cotton's producing a lot, but when you go to weigh it, it doesn't weigh very much.
I can tell you about one case last year. One of my brothers grew some next to my field. When it was weighed, he'd loaded up a 7-ton truck, but in the end, there were only 2.2 tons of cotton.
The best cotton is the one that weighs more. It lets you earn enough to eat. Anyway, I don't want GMOs. Absolutely not.
What are the reasons behind the lower than expected GMO yield? In Monsanto's opinion, it's partly due to the lack of training of small farmers who are often illiterate and do not correctly apply the complex procedures of GMO technology.
People need to understand that they've got a high-tech car which doesn't drive like a field car, a regular car. There are rules to respect. like proper fertilization. I'm just saying any old number here, but if you have to put 100 in fertilizer and you only put 50, well, obviously, you'll get the yield for 50 and not for 100. And treatment. You still have to do 2 treatments, even for genetically modified cotton. I'd be surprised if there weren't farmers who skip 1 or 2 to avoid paying for pesticides to save money.
In Burkina Faso, farmers are accused of diverting a part of the cotton fertilizer towards other crops. But the truth is, many farmers simply don't have the means to buy fertilizer, and they move into the cotton sector in order to acquire seeds and fertilizer on credit from cotton companies.
It's true that it's a way to get access to pesticides and fertilizers, and they use these inputs in their grain fields as well, mostly for sorghum and corn. Because above all, they need to eat. The farmer has to eat. And when you take small farmers, those who have 1, 2, 3 hectares of cotton, maybe 4, who really don't have huge surfaces for cotton growing, they really suffer from it. They have lower and lower yields every year. Most of the farms are family-run farms. We don't have huge cotton producers here, so it means that it's just for an elite.
There is indeed an economy of scale, which means that you amortize more easily with 150 hectares than you do with 10, let alone 2. Every large-scale farm in the world, and I won't specifically mention South America, but obviously there too, is well aware of this. $1 earned on 1 hectare multiplied by 100,000 hectares makes $100,000. And if you multiply it by 3 hectares, well, that makes $3. It's that simple.
But in Burkina Faso, it's not only the small farmers who are complaining about BT cotton. The cotton companies are too, because the quality of the fiber has declined in comparison to conventional cotton. The proportion of short fibers has increased, supplanting longer fibers.
The staple here is a bit shorter, isn't it? That's not good. It's very short. 1 inch, 1/16. It's always 1 inch, 1/16.
In 2015, faced with a decline in quality that was affecting the reputation of Burkinabe cotton on the international market, Sofitex decided to reduce the proportion of GM cotton in its overall production from 70 to 60%. Socoma, the country's second largest cotton company, majority owned by French group Géocoton, decided to go even further.
The impact of short fiber is equal to billions of CFA francs. I can assure you, as the CEO of Géocoton, that Burkina Faso cotton would be in real danger if we continued down that road. Because we had over 56% of low quality. At some point, the markets would react and move towards other origins. So yes, commercially, there was a real danger. There was a real danger, and we had to react. That's why we decided to reverse the trend. GMOs represented 70% of production last year. They'll represent 40% this year.
Monsanto appears to have a solution for this problem. A new BT cotton called Roundup Ready Flex, into which an additional gene has been inserted that makes it tolerant to Roundup. Currently being trialed in Burkina Faso's test fields, it would bring a return to a good fiber length. But some wonder if it isn't a strategy to increase the American firm's sales of Roundup in the country.
We met with Monsanto just 3 weeks ago. Things got pretty heated between us. Apparently, the length issue's been resolved, but it just so happens that it doesn't correspond to a demand. So we're not too keen on going down that path. It might be a business thing. They're businessmen.
Roundup Ready Flex. I think that's an avenue we'll pursue, yes. It's a business, eh? We all agree companies don't do charity. But yes, we're satisfied with what's happening. We're going to do more for Burkina Faso, obviously, and for the sub-region.
The director of Monsanto in Burkina Faso says that this glyphosate is a total pesticide. It kills everything, except BT cotton. And he's happy about it. That's what astounds me, that he's happy. We're going to kill biodiversity, and then what will become of us? It kills everything, it even kills us. And then we'll see. We could call Burkina Faso its open-air laboratory, because everything's being tested here, no holds barred. Other products have been announced, niébé, beans, corn, and those are daily foods.
And while GMOs do not always keep their promises to farmers, they still raise numerous questions for consumers. Including in the United States, the leading consumers of GMOs in the world. Here, the industry is waging a fierce battle to prevent the mandatory labeling of food products containing GMOs, as is done in Europe.
In the US, most of the studies suggest that it's somewhere in the order of 70 or 80% of processed foods contain some ingredients that came from a genetically engineered source. So that's the salad dressing. This cereal, wow, look at those colors. Okay, sugar, you don't even know what the sugar source is, so you don't know if it's engineered or not. Corn, that's almost certainly going to have engineered components in it. You know, we could go down the list. Almost all developed countries and many developing countries require labeling for genetic engineering. And in the US, survey after survey consistently show that 90% or more of consumers want to know. They want a mandatory label that tells them whether the food is engineered. So in a democracy, and especially for something as important as food, you would hope that consumers would have that choice. And in fact, there have been no epidemiology studies of the human population in the context of genetically engineered food. So when my colleagues say, and many scientists do, we have been eating this for 2 decades and it's never caused a sneeze or a sniffle, or it's never caused, that is, just junk science. You cannot know that without doing epidemiology. Absence of data is not evidence of absence of harm.
We know very little about the risks of consuming either plants that produce toxins against insects or plants that resist herbicides, which means that we're eating herbicides. When there were no GMOs, you couldn't eat Roundup because every plant that got treated with Roundup died. But the moment there are transgenic plants that resist Roundup, you're going to be eating Roundup. And to be sure that this poison, regularly consumed over large periods of time, has no effect on us, we'd need to conduct more thorough studies than what's been done up until now, which have been short-term, involving a few mice.
In September 2012, the publication of a scientific study exploded onto the headlines. Directed by French researcher Gilles-Éric Seralini, the study focused on Monsanto's NK603, Roundup-tolerant transgenic corn. And it raised serious questions about its safety. It was a first in science, the first GMO toxicity study conducted over 2 years. In other words, the entire lifespan of a rat. The photos of the tested animals swollen with tumors were seen around the world. Professor Seralini and his team called for an extension of the requisite test periods that European regulators require of GMO companies before giving market approval. Tests were being conducted over only 3 months. But Seralini, over a longer term, observed toxic effects on rats.
These were the results we got. First, a higher mortality rate in rats treated with GMOs and Roundup than in the control group of rats. Next, mammary tumors that killed up to 70% of females, which developed in the first year, compared with 30% in the control group, which developed at the end of the second year. Meanwhile, the males didn't die of mammary tumors, but they died of severe liver and kidney damage. In other words, the organs of detoxification were affected, the liver and the kidneys, which are the organs that filter food and drink. I found the results extremely worrying because the body is well made. It ends up detoxifying. It's not the end of the world, but the more you give it, the less it can handle. At some point it gives up and you get chronic disease, not just because of the GMO, but because of the pesticides it contains. In my opinion, it's not so much because the genome was fiddled with, which would be the initial superficial analysis, but because the genome was fiddled with for a specific reason, which was to turn these plants into pesticide sponges.
The instant it was published, the Seralini study provoked the fury of GMO manufacturers. It sparked enormous controversy, both among scientists and in the media.
Gilles-Éric
Seralini was attacked by sock puppets in an extremely violent way. He was accused of being a member of a vitalist Christian sect. He was associated with all sorts of anti-scientific movements, which was completely unfounded in my opinion. We have here another technique used by the biotech industry, which goes after people instead of results, the results being discredited along the way.
Les résultats étant bien évidemment discrédités dans la foulée. Monsanto
doesn't hide that it was an entire department of researchers, of scientists, who work at debunking or discrediting any science that might be troublesome for business.
We have proof that advertising agencies linked to Monsanto changed my CV twice a day on Wikipedia. And by that I mean people would find articles that said I was a fraud or a cultist. They said I'd committed fraud. They weren't the right rats. I shouldn't have used Sprague Dawleys. Why? Because those rats are apparently too sensitive, too prone to developing tumors. Monsanto used the same strain for its own trials. I took the rats that they used, and more importantly, the rats recommended by international institutions for these tests.
Mesdames, Messieurs, nous accueillons en audition commune le professeur Gilles-Éric Seralini et le docteur Joël Spirou, co-auteur d'une étude.
Faced with the controversy and growing public concern, politicians were forced to react. A debate was organized at the National Assembly. Committees of experts were called upon both in France and in Europe. In the end, the conclusions of the Seralini study were rejected, mostly because it was conducted with groups of 10 rats, while carcinogenicity studies are normally done with groups of 50 rats. And even if Professor Seralini hadn't expected to observe differences in tumor growth, his sample size was too small to draw valid conclusions.
Gilles-Éric Seralini's study does not allow us to conclusively say this type of GMO, associated or not with the herbicide that it resists, leads to the kind of pathology that was presented in the rats. It's possible, but it's equally possible that it was pure chance. In a word, the study's statistical power doesn't allow us to draw conclusions, which Gilles-Éric Seralini himself admits.
Of course there could have been more rats and more statistics. I'm not saying we wouldn't have liked to have done it on 20 rats or 30 rats, but we'd have needed not 3.2 but 6 million euros for all those tests. With the resources we had, it was a study that was the only one of its kind in the world.
Faced with the shockwaves created by the Seralini study, the French government and the European Commission eventually agreed to launch studies lasting longer than the requisite 90 days to analyze the effects of GMO consumption on health. In the city of Toulouse, the director of the Toxalim Research Center in Food Toxicology is coordinating GMO 90+, a €2.5 million project funded by the Ministry of Ecology. Trials began in August 2015 using the NK603 corn studied by Seralini and advanced omics analysis techniques like metabolomics to analyze the rats' urine samples.
The aim of this research project is to watch the rats over 6 months and to observe, both in the urine and in the organs, any potential changes either in gene expression or in the rat's general metabolism. We're sure that, statistically speaking, we have enough animals. We can't be attacked in terms of experimental design. Regarding the duration of the study, well, we can do 6 months. Why not longer? Well, we can do 2 years, but in that case, we're not talking 2.5 million, we're talking 5 million. What I mean is, there are contingencies. And on a purely scientific basis, since we're connected to other European programs, which are longer term, we'll have access to organ, blood, and urine samples on rats that have been fed for 2 years. So the results for them are early 2018. I don't know what the result will be. I mean, if I knew the result, we'd stop doing experiments, right?
French scientists are indeed participating in a European study called G-Twist, which examines the same corn over a 2-year period. The project is coordinated from Germany. Testing also began in August 2015, and the results won't be known until early 2018. Unlike his French colleague, however, the German toxicologist conducting the study seems less uncertain of the results.
My greatest hope is that with the G-Twist project, we will present a study that has been perfectly conducted, with data that may not please everyone, but is the result of an irreproachable food study. And given the methods that we used, I can already vouch for it. Even if, once again, these results will displease a large part of the public.
Professor Pablo Steinberg was born in Argentina. He's also the toxicologist in another project funded by the European Commission called GRACE. It includes a toxicity study of a different genetically modified corn, Monsanto's Mon 810, which is grown in a number of European countries, including Spain. In the fall of 2015, the conference presenting the results was attended by experts from 20 countries. They included many scientists, a few NGOs, and numerous agribusiness representatives such as Monsanto, Bayer, BASF, and DuPont. It's no coincidence that the German project coordinator chose to organize the event in Potsdam, near Berlin, on the 9th of November, the anniversary date of the fall of the Berlin Wall.
Ladies and gentlemen, dear colleagues, I will open the final press conference with the sentence by Erich Fried. Those who want the world to continue as it is do not want the world to continue. Für uns ist der 9. November
November 9th is an extremely important date for us because the fall of the Berlin Wall was a major event, not only for us Germans, but for the entire world. And we too, with our work, are trying to break down a few walls and get movement on certain fixed positions. That's why I find it very symbolic to have organized this conference in Potsdam, near Berlin, and the remains of the wall.
Regarding the one-year feeding trial, the feeding of maize Mon 810 to rats did not lead to adverse effects. We have had a number of so-called statistically significant differences. That means that the numbers of a parameter between two groups have differed, but we differentiate between simply a statistically significant difference and a toxicologically relevant difference. So even if we were seeing certain differences in relative organ weights, we don't think that that is of a toxicological relevance.
Well, someone in the audience who wants to comment. Yes, Bruno?
I think that we can use now omics technology to check if there is not a disease, but some imbalance in some organs. So when I look into the results, so I was really, I don't understand why so the the feeding with GMOs, they are smaller and they eat less. So we have to think about it.
In this study, which lasted 1 year, we have noted that in one of the test groups fed with genetically modified plants, weight gain was significantly lower, but we don't know why.
Scientists from the GRACE project can't explain why, over the long term, rats eat less when their food contains GMOs. But this, for them, is not a health issue. As we can see, in toxicology, it's the way results are interpreted that counts. And in this case, the stakes are high. Because the point is to determine whether or not GMO companies should be required to conduct studies that last over 3 months when they apply for market approval in the European Union.
So, ladies and gentlemen, now we come to the general conclusions and recommendations. There's been a long controversial discussion if we perform feeding trials and how long should these performed feeding trials be extended. Only for 3 months? Should we go then for a 1 year or even for a 2-year feeding trial? And the feeding trials did not reveal any scientific trigger for an extension of the feeding period.
Thank you very much. We have time for some questions, please.
When I looked at this Mon 810, I would say, well, we could expect some subtle effects which are not so easy to detect in 90 days. But when we look to the 60 events being authorized for import, we see a lot of quite more complex traits. Stacked events, which produce up to 6 Bt toxins, or events that can be expected to have residues from pesticides. We have to make sure that the process as such is not biased, is not driven by some networks which are very close to industry, and that we in the end really can trust the outcome. And this, I have to say, is not the case in GRACE, and that's our problem. I think this was an exercise with an outcome that was quite clear from the beginning.
So, I think that was a comment which we will not comment.
This is a huge problem for us, particularly the fact that Joachim Schiemann belongs to the ISBR, the International Society for Biosafety Research. He's even served as its president. It's a group that's present worldwide and whose conferences are sponsored by the industry. So he's more of a lobbyist than an objective scientist. A few years ago, he even filed a patent for genetically modified plants.
Das wäre natürlich sehr schön, wenn wir an irgendeinem.
Obviously, it would be great if there could be a new November 9th, if possible in the near future, so that we could put an end to this Cold War in the field of genetically modified organisms. Anyway, we will keep going.
Aber es geht weiter.
In response to concern, biotechnology research scientists are now pushing new second-generation GMOs, which they claim are better not only for farmers, but also for consumers. In particular, crops that have been genetically modified to enhance nutritional quality by being vitamin or omega-3 enriched. One of these crops is particularly controversial, golden rice. A rice designed to fight a public health problem in low-income countries, vitamin A deficiency, which weakens the immune system. Close to 700,000 children die from it annually, and over 300,000 become blind. Golden rice is currently being tested in Bangladesh, Indonesia, and most of all here in the Philippines.
IRI is a humanitarian organization. We do research for popular and people's good. The idea is that we're very concerned, as many people are, about vitamin A deficiency. Almost 200 million people are affected. So the idea is, if people like rice and they eat a lot of rice every day, why don't we improve the nutritional content of rice?
Golden rice is more nutritious. It's very yellow because it has beta-carotene in the grain. So when you eat beta-carotene, either from vegetables or from rice, your body processes beta-carotene and converts this to vitamin A as the body needs it. The addition of two genes, one from maize and one from a common soil bacterium, enable the plant to produce beta-carotene in the grain. We do the breeding here. If you see these plants, we are introgressing the vitamin A genes into an Indonesian variety, Cerang.
Research into golden rice began in 1990 in a greenhouse of the Swiss Federal Institute of Technology in Zurich. 9 years later, after numerous trials, the first prototype of golden rice was created. To his great surprise, its inventor made the cover of Time magazine.
When my colleagues saw this photo in Time magazine, they said, "You'll be getting the Nobel Prize in a couple of years." It was the first time a botanist had made the cover of Time. People understood that this was something big. In fact, 6 days later, Monsanto called me. I was very surprised, but we came to an agreement very quickly. They owned one of the patents for which we wanted a free license, and they spontaneously offered to give it to us, this license, right away. Some of them said, Monsanto wants to steal the limelight of this project.
The GMO giants like Monsanto in America and Syngenta in Switzerland strongly supported the development of golden rice, which Professor Potricus has declared he wants to distribute freely to small farmers. But some people are fiercely opposed to it, in particular the environmental organization Greenpeace, because golden rice would serve most of all to gild the image of GMOs.
For Greenpeace, the publicity, especially around golden rice, is for it to be, to increase or to gain acceptance for golden rice and including GE crops, other GE crops in developing countries like the Philippines. The multiple patents for golden rice are still held exclusively by Syngenta and Monsanto, although including any improvements to this technology. However, Syngenta did say several years ago that they have no intentions to commercialize GE rice, GE golden rice, but on the other hand, there's also no legal obstacle for them should they decide otherwise in the future. And then the cycle of dependency to this rice variety will become quite irreversible.
We feel in Masipag that farmers will lose their rights over their seeds because once these seeds is contaminated, once these seeds are lost, they cannot be retrieved anymore. The contamination would come from seed mixing, actually. For example, golden rice would be accidentally through post-harvest processing, like threshing. It could be mixed with ordinary rice. So we don't want to lose our traditional rice varieties. That's why we are fighting this GMO.
In the Philippines, where rice is a staple food, the Golden Rice Project had emotions running high. On August 8, 2013, a group of protesters destroyed one of the 5 test fields.
I was outraged, not just saddened, but outraged. This photo was taken in the Vatican during a meeting of the Pontifical Academy of Sciences. At the end of the meeting, I asked the Pope if he would agree to bless golden rice, and he did. I hope that will help golden rice to be more accepted. This is the sample I showed him. Fresh golden rice has more color, but this is 3 years old and it's faded a bit.
Golden rice was, probably, done by some public interest scientists who are very well-meaning. But now there is data emerging that suggests that the beta-carotene may break down in storage too quickly. Okay, so some other scientists have said the problem is that now the golden rice varieties, the types of rice that the golden rice is in, are not productive enough. So you can't give it to farmers if the rice is not going to be productive. So that's a technical problem, and they're trying to work on that. So we're talking about a project that's already 25 years old. Lots of time and money of effort of scientists and others for developing the first generation, which didn't produce enough beta-carotene. And then they had to re-engineer it to produce more, and it's still going to be a minimum of a number of years before it gets out in the field. Is that really a cost-effective way to deal with this problem?
There are two solutions. An agronomical solution that makes sense to me would be to say, okay, let's stop giving kids only rice. Let's grow a few eggplants on the edge of the plot and give them eggplants and rice together. That would be better on many levels and, among other things, would bring them the vitamins they need. That's what I call intelligent agronomy. But it's not technological in the traditional sense of the word, so it doesn't appeal. To sound modern, you can't put eggplants with rice. You have to make rice with eggplant genes inside. Frankly, for me, that's not an intelligent solution. That's a stupid use of technology.
Can GMO technology prove to be useful for other public health issues? Some scientists use it to fight illnesses like dengue fever. This tropical disease is rarely fatal, but can be very crippling, causing high fevers and affecting over 50 million people every year across the world, particularly in Brazil. Scientists here are not working on plants, but on genetically modified insects to try and eliminate Aedes aegypti, the terrible mosquito that transmits the dengue virus. A transgenic mosquito has been developed by British company Oxitec, which opened a factory in Brazil capable of producing several million eggs per month. Scientists took first-generation eggs and injected a lethal gene into them. This gene produces a protein that makes the mosquito dependent on a yellow-colored antibiotic, tetracycline. Scientists raise the larvae by providing this antidote in the laboratory. Once they're released into the wild, however, the mosquitoes, deprived of this substance, die within a few days, as do their descendants.
It's a very, very simple idea. We send out males, they carry a gene that when they mate with females, the offspring don't survive. So every female that mates with one of our males, their offspring die. And if you can put out enough males, you'll bring that population right down. We've done several trials in Cayman, Panama, Brazil, and in each one, the results have been very, very consistent, which is 90% or more reduction in about 6 months. I would say it's a complementary tool. People are already using insecticides, pheromones, sticky traps, anything they can to try and control the insect, but it's not enough.
On April 30th, 2015, Oxitec scientists launched a new program in which they released around 1 million transgenic mosquitoes per week for 10 months in a neighborhood of the city of Piracicaba, near Sao Paulo, where dengue is prevalent.
We spend over 1.3 million euro a year to fight this scourge. An efficient scientific breakthrough in this area, preferably at minimal cost, if the company lowered its prices, would represent real progress for Brazilian society. That's all. Thank you for coming.
The major inconvenience of this technology for a municipality is that you have to constantly renew the process. When transgenic mosquitoes stop being released, nothing prevents wild mosquitoes from neighboring areas from returning to the treated zone. On the other hand, this constraint could be very lucrative for Oxitec. They're impatiently waiting for the Brazilian authorities' response to their application for market approval.
It'll be a big thing for us. At the moment, we can do projects, but we can't go out and we can't make claims. So that will be a key approval, because at that point, we can then go to other municipalities and say, would you like to do the same thing as Piracicaba? Do you want to do this all year, or do you want to do it just before the rainy season and during the rainy season? It entirely depends on the objective. It can be used in different ways.
In 2015, the Brazilian health authorities gave preference to a different technology: vaccine. The first vaccine in the world against dengue, developed by French laboratory Sanofi Pasteur. But research projects on GMAs, or genetically modified animals, are far from over. There are more and more of them, and they target even more species than for plants, including flies, mice, pigs, rabbits, or goats and ewes, whose milk produces molecules for medicines. In the Argentinian province of Buenos Aires, scientists have created a cow that's absolutely unique, which goes by the sweet name of Rosita.
Quieta! No, no, no! No, 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 no. Oh, oh. Ay, me pisó.
After a transgenesis and a cloning, the cow was born on April 6, 2011, to produce milk with the same qualities as human mother's milk.
She's a Jersey cow. The scientific importance of this cow is that, for the first time ever, we've succeeded in transferring, in just one go, 2 human genes into a bovine genome. These genes produce 2 proteins, lysozyme and lactoferrin, which play a vital role in breastfed babies by boosting the immune system and encouraging iron absorption. Our goal is that one day this animal, and the animals conceived by her, will produce milk that can answer the needs of babies who can't be breastfed. But it is a very long-term project. There are still many tests to be done and numerous challenges to overcome. The idea is to obtain an animal capable of producing more transgenic proteins and to eliminate certain other proteins, like the ones that cause milk allergies.
Today, Rosita lives in an isolated pen, protected by laser sensors and an alarm. Genetically modified animals are enveloped in safety standards that reflect the fears they generate. What would happen if they escaped into the wild, reproduced with other animals, and passed on their transgene? That's what long blocked the market approval of another transgenic animal, AquaVantage salmon, whose stated advantage is to grow twice as quickly as ordinary Atlantic salmon and therefore spend half as much time in fish pens. A fish that's cheaper for producers and perhaps cheaper for consumers. It was created in 1989 by Canadian research scientists, then developed by the American company AquaBounty. Genetically modified salmon are today conceived in Canada, then fattened on fish pens down in Panama. They're confined to the middle of the forest, far from the ocean, to limit the risk of dissemination and crossbreeding with wild species.
I'm actually quite confident in the regulatory structures that I've reviewed for Canada and the American government. There is very, very little likelihood that these genetically modified organisms will ever have the opportunity to interact with any natural populations of fish, never mind Atlantic salmon. They have to be produced in land-based facilities that are I want to say escape-proof. Obviously, nothing is 100%, but the probability is very, very, very low. So you have physical containment of these organisms, but there's also a biological containment measure. These organisms that are going to be produced are being produced entirely as all females, so that they could not reproduce if they were to escape. But on top of that, there has been a manipulation that makes them sterile, so they couldn't reproduce anyway.
But concerns for the environment persist. But the sterilization technique is not completely efficient. What if genetically modified salmon were to escape into the sea, following some natural disaster that destroyed the facilities, for example? The effects would be impossible to reverse. Independent scientists have also studied the risks associated with this transgene. It was designed with genes from 2 other species of fish, which boost the production of a growth hormone all year long. The result? A transgenic salmon that is voracious, aggressive, and seems predisposed to developing skeletal deformities as well as certain diseases. Its numerous critics have baptized it Frankenfish.
What we know about this growth hormone, it's called IGF-1. We know it causes breast cancers. We know it causes lots of other cancers. Are you going to eat enough salmon that the additional IGF-1 will, in and of itself, cause cancer? Probably not. But why introduce into our diet another thing that adds to that cancer burden? In the years that they've been working on this, there's a Norwegian company that through traditional breeding can get salmon to market size just as fast as this genetically engineered salmon can. So if you're a French consumer and you have the choice of a GMO salmon with more growth hormone than normal, or a Norwegian salmon that's been traditionally bred, what are you going to choose? You're going to choose the fast-growing Norwegian salmon, but not the American with growth hormones. So, I mean, it's a bad strategy economically, and it's a lousy food for people.
Despite concerns, on November 19, 2015, American health authorities ended up approving the marketing of this fish, the first genetically modified animal authorized for human consumption. And, as for other GMOs in the United States, there is no mandatory labeling that would allow differentiation between genetically modified and traditional Atlantic salmon, like the one sold in this Canadian supermarket. For consumers, therefore, it's impossible to know exactly what they are buying and what they will be eating.